Well, biosimilars are approved by the FDA based on a different mechanism than the original products. So the original biologics, some of which go back 20 years, were approved as an originator, an innovator drug. They had to provide not only the preclinical data, but several randomized controlled trials demonstrating efficacy and safety. Obviously, that's an extremely costly process, and to facilitate the development of biosimilars, a more expedited process was put in place by the FDA a few years ago, which put much more emphasis on the molecular characteristics and structure and its biologic behavior in the patient, to pharmacokinetics, pharmacodynamics, whether there's any antibodies formed, immunogenicity, all these things to demonstrate that it's very similar, highly similar to the original product, but without the necessity of going through the multi-million-dollar large randomized phase 3 trials. Now, some of the biosimilars have done that anyway, but the idea here is to reduce the cost of development so that that cost reduction could be passed on in terms of lower pricing, greater competition in the marketplace. So it's really a focus on the preclinical work and the safety in patients, and the assumption that if it's the same, virtually the same molecule, same mechanism of action, doesn't have any safety concerns, it will function essentially the same as the original biologic product.